I would like to say welcome, everyone. So I'm your host for today, Martin Extekate, and I'm at the Heinrich Heine University. Today is yet another episode in the so-called Interdisciplinary Lecture Series that are being organized by our Marie Curie doctoral networks, ComIncel. And today the speaker will be Professor Rumiana Dimova. So she is from the Max Planck Institute of Colloids and Interfaces in Germany, where I'm also located. And she is also the head of the ComIncel that we are currently part of. And today she will talk about phase separation in biological systems from membranes to biomolecular condensates. Rumi, please go ahead.
Thank you, Martin, for this nice introduction. And because today is the 4th of May, May 4th, may the force be with you, if for those of you who are fans of Star Wars. And here's a version of the title adapted to the Star Wars language of Yoda. May the phase be with you. Phase separation everywhere it is. Condensates and membranes connected they are. So if this does not make sense to you, I hope by the end of the lecture that it will. What you see on this slide are membranes of giant vesicles in red and condensates that can be either inside or outside. But let's start first with the membrane. This is the outline of the lecture. I'll first talk about phase separation in the lipid membrane. Then in the cytoplasm, outline a couple of techniques to study condensation, and then come back to membranes and condensates, putting them together. So when you think of phase separation in the membrane, you have to think of what do lipids do. They undergo a phase transition under certain temperatures. At very low temperatures, they're in the gel or solid ordered phase. At high temperature, they're in the fluid or liquid disordered phase. In between, there could be also a pre-transition called also rippled phase. So this cartoons practically illustrate the state of the tails of the lipids. And then you can also talk about fluid liquid ordered phase when you add cholesterol. To understand the difference between ordered and disordered, liquid ordered and liquid disordered, which are the most relevant phases in biological membranes. Here's another cartoon. And just a disclaimer, these hand-drawn cartoons are practically part of this review that was recently published, but they were redrawn by the journal office. These here were painted by my, or colored by my daughter. Here, the difference between liquid ordered and liquid disordered, as I said, these are probably the most important phases in biological membranes. Why are they called liquid ordered and liquid disordered? They are liquid because when you look from above, the head groups don't have any order. They are fluid. These head groups can move. The lipids can move relative to each other and diffuse. But then when you look from the side, the ordered phase has more ordered tails of the lipids compared to completely disordered tails in the liquid disordered phase. Why are we talking about these two phases? Because they are very characteristic also for biological membranes, which are not homogeneous. You can have phase separation in them because they are multi-component. And the Raft hypothesis simply tells you that there might be liquid ordered domains in a liquid disordered environment. They can be very tiny, submicroscopic, and certain proteins partition into them. So these are called lipid rafts. This is a picture generated with ChatGPT. I don't know what these things are here that ChatGPT imagined, but you have a very large variety of components. And these lipid rafts are important for processes like signaling, trafficking, and curvature. Now, the membrane phase states and membrane phase separation can be represented by understanding phase diagrams, and I'll come to that later. But the way you can visualize these phase separation processes in a very convenient way is by using an excellent membrane model, which are giant vesicles. Images of these are shown here. Why is it important to study model membranes? It's because we can isolate physical principles from biological noise, where in biology we have all sorts of active processes taking part, or multi-component systems. Also, with these model systems, you can look at the coexistence of liquid-ordered and liquid-disordered membranes, and you can precisely control the membrane composition, and the conditions are well defined. You can then examine the effect of temperature or other factors. Here, what you see are vesicles exhibiting phase coexistence, and the way this is done is by putting tiny fraction of labeled lipid that partitions in one domain, and then the red one partitions in the other domain, so liquid ordered and liquid disordered, and then you have two different compositions where the liquid ordered phase is much larger than the liquid disordered one. Now, this slide introduces the miscibility transition in terms of temperature, the miscibility transition temperature T mix. Above this temperature, the membrane is completely homogeneous. This is how a vesicle will look like. And then below, you can have phase separation, and the domains are forming that they can coalesce and coarsen over time. So the message here is when you have cooling, you can induce phase separation. How does it look like in terms of quantitative description of the system? Let's first start with a binary mixture, so two-component membrane. A membrane composed of lipid 1 and lipid 2. Lipid 1 melts at high temperature, so above this temperature, on this axis you have temperature, so above this melting temperature, the lipid is in the liquid disordered phase, below it's in the gel phase, for example. And the same thing for lipid 1, so 100% mixture is located here. Here you have 100% of lipid 1. And in between you have a mixture of the two. At very high temperatures, you have a homogeneous phase, liquid, and at low temperature, you have again a homogeneous membrane that is in the solid phase. In the middle, you have something called coexistence region, where domains would be observed, as sketched here. And these black lines are called binodals. They separate the single phase region from the two phase region. This is the two phase region. And then if you take a point somewhere here, so this point is at certain temperature, and the composition is read by this axis. If you take this point, you can look at something called a tie line, which is in this case a horizontal line that connects your point with the, it makes a cross section with the binodal. The cross section with the binodal, so these little points, would tell you what the composition of the individual phase in your vesicle are. For example, this would be the composition of the purple domains, and this would be the composition of the yellow domain in your vesicle. So it connects compositions of phases that coexist in equilibrium. Tie lines are good to know because, in addition, if you have a tie line around at your point of interest, there's also something called the lever rule, which tells you that the length of this segment here versus the length of the rest of the segment of the tie line is inversely proportional to the area ratio of these domains. So the solid domain, which is located here, has a smaller fraction than the liquid domain located here, because you have a different ratio of this segment lengths. So the lever rule predicts how much area each domain type occupies practically. That's why it's important to know these phase diagrams. Okay, I have two slides about ternary mixtures. So now we're complicating the mixture. So we have now three-component mixture. And the way you plot it is you plot this phase diagram for a fixed temperature and this is a triangle now, not a line like here. and here you have temperature. You have a line here between the two lipids. Now you have three lipids or three components. At the tips of the at the edges of the not the edge, the ends of the triangle, you have the 100% of each component, and the shoulders are the binary mixtures. A and B are located here, and the three component mixtures are located somewhere here. And how to read these compositions? At the line which is horizontal, you would have all of these compositions that are this line would have 20% of component A, and then you go up. The same thing you can do for B. So now if you have a three component mixture, for example, located here, like plotting such lines parallel to the shoulders of the triangle, you can practically read the composition of each three components. And here's how an example of a ternary mixture may look like. If you have a lipid 1, which has a low melting temperature, lipid 2, which has a high melting temperature, and then add cholesterol. First, there is a region where you cannot make membranes because sterols form crystals. And then you have a bunch of different regions. Again, the black lines are the binodals. You have a region of homogeneous liquid ordered phase or liquid disordered phase or solid phase. And then you have regions of two-phase coexistence, liquid and solid, and liquid disordered plus liquid ordered, which is the most interesting one. You can have also a three-phase coexistence region. These are just a couple of examples of vesicles that are phase separating in this region. Here you have a vesicle with a small fraction of liquid ordered domains in a larger fraction of liquid disordered domain. And here you have the inverse situation where the liquid disordered domains have a much smaller fraction. With increasing temperature, so as a reminder, this phase diagram is at a fixed temperature, but if you start increasing the temperature, and you can plot this phase diagram by plotting, by having an additional axis which is perpendicular to the screen, which would be your temperature. So if you start increasing the temperature, these domains would practically start disappearing one by one, and then you are going to have a homogeneous, they would shrink. The regions would shrink, and you would end up with a homogeneous membrane. This is how real images may look like. And from the shape of the domains, you can guess where in the phase diagram you are. So there are two lipids and cholesterol. Homogeneous membranes look kind of boring. You don't have anything on them. And then liquid ordered, liquid disordered. You have domains which are round. So fluid domains have smooth boundaries. Solid ones don't. Here are examples of solid domains. And you can resolve them by using fluorophores that can partition differently in phases. Here, in this case, only one fluorophore was used. But you might be able to see that in this particular example, you have three types of domains. Maybe you distinguish the bright one, the less bright ones, and then there are very black ones, which are the solid ones. So solid phases typically exclude fluorophores. Another feature is that fluid domains can coalesce when they meet. Solid ones would cluster, would preserve their shape. And then also solid domains can show up as facets or flat parts on the giant vesicle. So here you see that the membrane is kind of a different shape that has different protrusions. Okay, so the mechanism of phase separation. The slide now introduces these two mechanisms, nucleation and growth, and spinodal decomposition. In the case of nucleation, you must overcome a free energy barrier, and here small nuclei appear and then they grow over time. While in the spinodal decomposition, the fluctuations are growing spontaneously into interconnected patterns that appear and then spontaneously break into domains. Here are examples of this occurring on giant vesicles. So domain, here we have coalescence, so following this nucleation and growth can proceed via coalescence or austral ripening. I'm going to introduce this a little bit later. where you just see the formation of the domains that can either coalesce to grow to make larger domains, or can undergo Ostwald ripening, where the small domains disappear because material from them is diffusing to the larger domains driven by line tension. Spinolo decomposition example is shown here, where you have the interconnected patterns appearing and coalescing over time. And there is also something called viscous fingering, where you have very low line tensions and the domains are no longer circular, but have different distorted shapes. So now I was talking about model membranes in general, but from model membranes to cells, you can still think that phase separation principles apply as well, even though you have proteins, cytoskeleton, and active processes. And I'll just give here two examples where such microscopic domains as those observed in giant vesicles have been observed in real plasma membrane systems. What you see here on this snapshot is a cell which is exposed to a cocktail of chemicals. It produces something called a giant plasma membrane vesicle, or a bleb. And if you change the temperature, this bleb would, or giant vesicle would undergo phase separation, just like we observed phase separation in giant vesicles. So despite the membrane complexity and the proteins and the active processes, phase separation can be observed in vivo. And here's one more example of micron scale domains observed in a membrane organelle called vacuole in the yeast organism. And you see the domain formation here. So the main message is that phase separation can occur and can be observed. It's typically submicroscopic in cells, simply because you have a larger compositional diversity in this, not only three lipids, for example, but thousands of lipids. So with this section, the key takeaway said that phase diagrams can predict compositions and domain fraction. Dynamics can proceed via nucleation and spinodal decomposition. And I showed some examples of phase separation in real organelles. And now we go to phase separation in the cytoplasm. So this was two-dimensional phase separation in the membrane. Now we go to three-dimensional phase separation in the cytoplasm. The same physical principles apply here. And why are we talking about phase separation in the cytoplasm? Because it happens and it produces membraneless organelles or condensates. Now the cytoplasm is crowded. Here's simulations representing this because the macromolecule concentration is very high. And this crowding can lead to phase separation following the Gibbs phase rule. This example is probably very often shown on conferences for P granule formations in this cell. They are labeled in green, and you see that some of them would grow over time. And these are actually droplets that are not bound by membrane. They're just droplets in the cytoplasm. Now, these condensates undergo, so they form via phase separation. And as a comparison with the membrane, as I said, membrane, you have two-dimensional phase separation. Here you have two-dimensional diffusion. In the bulk or in the condensate, you have three-dimensional diffusion. Here, the shape of the domains is defined, and the circular boundary is defined by so-called light tension. Here, this is defined by the interfacial tension that defines the spherical shape of the condensates. Both systems evolved to minimize their interfacial energy. That's why they're both like circular or spherical in this case. Now, with the next couple of slides, I'm going to review the types of liquid-liquid phase separation in the bulk. The first one is segregative liquid-liquid phase separation, or also called coarservation, where components, for example, the two different components prefer their own environment. You have a blue and the green polymer, and then they phase separate, forming a phase rich in one of them, and the other phase is rich in the other one. So they are repelling each other, which is the driving force. Like liquid and oil are repelling, but in this case, you don't have liquid and oil. You have two aqueous phases. So both of them are made of water and one of the polymers. An example of such phase separation is the aqueous two-phase system, or ATPS, of PEG and dextran. And here's the example phase diagram where you have on the axis the concentrations of the two proteins, and the binodal divides the phase diagram into a region where you have two phase regions, so you have phase separation, and one phase region at low concentration. Here's one example for phase separation and PEG and dextran. If you put them in a test tube and they phase separate and you put some color, you're going to really see two aqueous phases, one rich in dextran, which is the heavy one, and the other one rich in PEG. And under the microscope, you can see also these droplets if you dope the system with a little bit of a fluor for that phase that partitions preferentially. The second type of phase separation is associative. It's also called complex coercivation, where you have, again, the two macromolecules, but then they like each other, typically driven by electrostatic interaction, and form a dense phase, which is rich in both polymers or macromolecules, and dilute phase, which is depleted. This is a key concept in biology. Most of the time, biological condensates are formed because of associative liquid-liquid phase separation. The phase diagram can be represented like this. Typically, phase separation, in this case, might be triggered by changes in salt concentration, pH, and the tie lines are actually horizontal. There is a mistake in this publication because you cannot have two phases at different temperature, for example. One phase at one and the other one at the other temperature. Examples are polylysin and polyglutamate coercivates, and this is a system, in particular this one is that is studied for a long time, already in 1957. It's tunable by salt and pH, and the coercivates show liquid-like behavior. And the final one is self-coercivation, also called simple coercivation, where you have only one component at dilute solution, it's one phase, but then when you increase the concentration, it can have phase separation, and this phase separation can be triggered also by pH and salt. And in this case, you have two phases, macromolecule poor and macromolecule rich phase. The phase diagram can look like this. You have a upper miscibility point, a lower miscibility point, and it's also called a reentrant phase, where at high salt, for example, concentration and low salt concentration, you have homogeneous phases, but then in between you have this condensate formation. It can be triggered also by pH and ionic strength. What is the driving force in biology for condensate formation? The core components are intrinsically disordered proteins. So these are proteins that lack 3D structure stability. And then the interactions between them are also characterized by multivalency, which means that they form many weak bonds per molecule. These interactions are also reversible. And the interactions are weak and transient, like pi-pi stacking of aromatic residues. It can be also electrostatic forces, hydrophobic interactions, and the energetic is such that these are attractive interactions that should overcome the entropic cost of concentrating the molecules. There's also something called the sticker and spacer model, which defines sticker interactions that lead to nucleation, or spacer that leads to gelation, or which is practically the aging of such condensates, a process via which the viscoelastic properties are changing. Similar to membranes in 2D, here again, the mechanism for phase separation is similar. This can it can follow a process of nucleation and growth, as sketched here, but this time we don't have two-dimensional domains. We have condensates or spinodal decomposition, where you have spontaneous amplification again of the fluctuations and interconnected patterns then form the condensates. The kinetics depends on how deep into the phase diagram you change. This is called a quench depth, so how deep, how much you change the conditions for phase separation. And growth can again proceed via coalescence of smaller domains into larger ones, or oscilloripening, where material from the smaller condensates diffuses to the larger ones, and then the smaller one disappear while larger ones are growing. High surface tension favors coalescence and oscilloripening. Low surface tension means slower equilibration and ripening and fluctuations. So again, a closer look at these phase diagrams, particularly for associated and simple conservation. On the axis, you can have a protein concentration, for example, and then if you have a certain point of interest where you are, where you have condensates, the tie line would tell you what the composition of your condensate is, read from the cross point with the binodal. So the green condensate would have this composition, while the dilute phase would have this composition, as you read it from this axis. And then I also wanted to mention the concept of hollow condensate. If you start, for example, here again, point 1, you have a homogeneous phase, you either decrease the temperature and go to point 2, where you have this phase separation. Now from point 2, if you go back or you do a little quench to point 3, instead of reducing the size of these domains, you can actually have phase separation inside the condensate, which this movie shows here. It is sped up, but practically inside the condensate, you have phase separation that leads at the end to a hollow condensate. It's hollow in the sense that inside it has a dilute phase, which is also the one outside. Now, I added these slides because some of the students in Common Cell are interested in measuring binodals in their phase diagram, or understanding their binodals. The way you can measure binodal is, for example, in the ATPS, in the peg-dextran aqueous two-phase system, is by doing cloud point titration. What you do is you first prepare highly concentrated solution of dextran and of PEG, maybe located here and here. And then you start in this point, you start drop-wise adding PEG, moving along this line. And the moment your solution, so here you start with the homogeneous solution, the moment your solution becomes cloudy or turbid, this is the point where you are crossing the binodal. So you write down this point, you continue it further, and then you start adding water. When you start adding water, you are moving along this line, which actually should meet the 0 of your phase diagram. And from this phase-separated system, you're again going to reach a cloudy solution, which would be again a point from your binodal. And if you go like this several times, you're going to practically resolve this binodal. Then you can, of course, knowing what you are mixing, you can convert the masses into weight percent and determine each point. And then you can, of course, choose a point somewhere here just to determine, to confirm whether you have a phase-separated solution as a verification. However, you have to remember that you have to equilibrate, leave the system to equilibrate for hours to define this. Another way of resolving a phase diagram, and this is an example for a system that undergoes self-conservation, is using turbidimetry and microscopy. So I measure the absorbance by increasing, for example, this is the phase diagram of glycine. By changing the salt concentration, the turbidity increases, and you see it with the absorbance, and then you detect this point and this point at high and low concentration. For example, this would be these 2 points. And then by changing the protein concentration, you can build the phase diagram. And in this region, when you put this solution under the microscope, you're going to see this droplets condensate formation, while here this would be a homogeneous solution, and you're not going to see anything except just diffuse. Okay, I I think I'll skip this, how to measure tie lines. And we'll just conclude this section by saying that liquid phase separation occurs also in the cell. An example for such condensates are organelles, membranous organelles such as nucleolus and P-body stress granules. These condensates in cells are dynamic, then sensitive to concentration and environment, and can mature, like age, over time. And this is one example here of the G3BP protein homogeneously distributed in the cells, but then when you're exposed to heat shock or chemical shock, condensation appears. And with this, I come to the next part, which is techniques that can be used to study condensates. And I'll mainly focus on model condensates in vitro. Here's an overview slide of 6 different techniques. On the following slide, I'm going to focus on condensate coalescence, micropipette aspiration, and trap. And here, just to give you an idea, one could use the sessile drop method, where you have a condensate sitting on a substrate, and you can visualize the shape of the condensate, which is deformed by gravity. And if you can do that, either by looking from the side view or doing confocal cross-section, vertical cross-section, if you fit the contour of this, the theoretical expressions for the shape of this droplet, your only fitting parameter would be the interfacial tension. For example, I'll give some example references where you can read further about these methods. The other one is passive microreology, where you take a little small particles that partition into your condensate, and then you track their Brownian motion. If you then plot the mean square displacement as a function of the different time intervals, you are practically going to learn something about the viscosity of this condensate. So slower movement means, a slow beat means high viscosity. And then you can do also active microreology, where you have beads incorporated into your condensate. They are manipulated with optical tweezers. And then when you move the position of one of the beads, you oscillate it and see how the force exerted on the first bead changes. You can practically measure the viscoelastic moduli G prime and G second prime, which is the storage modulus and the loss modulus. Each of them give you information about the elastic or solid-like properties and the viscosity of these condensates. And now a little bit more about condensate coalescence, which is a very popular technique to study interfacial tension and viscosity of condensate. The concept is very simple, and this is also illustrated in this movie. You put your sample on the microscope, and then you check for which condensates are coalescing, and you record their practically the aspect ratio of the resulting droplet, A over B, for example. Then you plot it over time. You fit the decay time. You deduce the characteristic relaxation time. And then do it for different condensates of different size. It's for this technique, it's important that the condensate that are coalescing have a similar size so that the theoretical description works. Here are examples of 3 different condensate couples coalescing and their aspect ratio plotted as a function of time. And then these fits here, then you get from that the relaxation time. This relaxation time is plotted for different couples of coarservates with different diameters, and the slope gives you the so-called inverse capillary velocity, which is the ratio of the viscosity and the interfacial tension. If you have a way to reduce the interfacial tension, then you can get the viscosity, which for this particular case is roughly 1,000 times more than the viscosity of water. Micropipette aspiration is another method to measure interfacial tension of condensates. So you apply some suction pressure and to see how the condensate deforms. The output is the interfacial tension, which depends on the size of your pipette and the droplet radius. And if you apply, if you change your pressure and you look how the response of the condensate is, so this length of here inside the pipette, which would be a little bit dynamic, you can get information about the viscosity of the droplets. This method can be applied both in vitro as well as in living systems. Here are some example references. And the last example is FRAP, fluorescence recovery after photobleaching. You can use a fluorophore that partitions in your condensate and then bleach a spot in your condensate and then monitor the intensity of this spot over time, which is plotted here. It recovers. The recovery time would give you information about the diffusion of your fluorophore inside the condensate. You can even use a diffusion coefficient if you have a decent recovery. Another output parameter is the so-called mobile fraction or immobile fraction, which is the one that is left over here. So if you don't have a full recovery, this means that some molecules are immobile, which become even more dramatic if your condensate is aging, for example. If you have complete recovery, this is more liquid-like behavior. If you have only partial or slow, this is the condensate shows a gel-like behavior. And again, aging reduces the recovery. And this, I like this plot from this paper because it shows you that depending on what you're working with, your system can be exhibiting very different viscosities. You see orders of magnitude here for this biological condensate. Here data for pegdextron system can be somewhere here, and also as in terms of interfacial tension, again, you can have a very diverse, very large range of interfacial tensions. So it's good to know where you are so that you know what to expect in terms of the dynamics of these condensates. The key takeaways, interfacial tension can be measured with pipettes or with the sessile drop method. Viscosity can be assessed from coalescence or microrheology. And you can learn about dynamics with FRAP. And now I come to the last part, which is the condensations at membranes and wetting. And this is something that my group has been working in the last few years, and we're very excited about that. To start with, here are some examples in cellular systems where condensates are interacting with membranes, like stress granules interacting with lysosomes, storage vacuoles interacting with the membrane of the vacuole, etc. Also tight junction. This is in this nice review by this group here in small methods. And in our group, what we are trying to understand this wetting and these interactions of condensates and membranes. And the way we do it is by using giant vesicles and protein-rich condensates. And if you bring these two, objects together, if they don't interact, you can talk about something like complete dewetting, which can change dramatically if you're changing the conditions in your system. So here we were changing the salt concentration. So you go from complete dewetting to partial wetting, where the condensate sticks to the membrane, to complete wetting, where it completely spreads over the membrane. The way to characterize this wetting or stickiness is by measuring a contact angle. This would work only if you have a membrane which is completely flat. But the thing is that with giant vesicles, because they are very soft and they can be deflated, they can observe different shapes, and it's not sufficient to measure just one contact angle. You should also measure all the others. And I'll tell a little bit more about this, but these two pictures illustrate a condition where you have in the same sample, the same membrane composition, the same condensate composition, but exhibiting two different contact angles. Now, what you also see is that the membrane here seems to be exhibiting a kink, or here, And this should not be possible because kink means an infinite bending energy. And if you did have a kink here, the membrane should practically rupture, but it doesn't, which means that this kink is not a kink, but the membrane is smoothly curved. And if you know this curvature, you can actually measure the real contact angle, which would be different from the microscopically measured one. This intrinsic contact angle is the one that characterizes the stickiness, while these ones would depend on the size of your vesicle, the size of your condensate, the tension on the membrane, etc. And just to convince you even further, with super resolution microscopy, we were able to resolve that this kink is not a kink, but the membrane is smoothly curved with the curvature of the order of 140 to 200 nanometer. Now, with the next slide, I'll show the only slide with formulas, but I thought that it's kind of important to resolve this thing and to describe this how to quantify the condensate affinity to membranes. We start with a vesicle that is wetted by the condensate, and we focus on this three-phase contact line. So it's a contact line that goes around here. At this contact angle, if everything is equilibrated and it does not move, the forces acting on it should equilibrate. What are the forces? You have the interfacial tension acting in this direction. This is the surface of the condensate. It's characterized by this interfacial tension. And then you have two membrane segments that compose your vesicle. One segment is the one to which the condensate adheres. And the other segment is the one to which the external solution or the depleted phase adheres. So they're characterized by membrane tensions, or labeled here. As I said, the force should balance here, which means that this vectorial sum should give 0, which also means that if I translate this vector here, and this one here, and this one here, they should form a triangle. This is also called the Newman triangle. The angles are kept, and this would allow you to write this geometric relation between the tension and the macroscopic contact angles. Now, if we look into these two tensions here acting on these two membrane segments, so this and this, you can also think of them as composed of a lateral stress, which would tell you how tense the whole vesicle is, and contributions from two kind of what you can call adhesion energy per area. So the adhesion energy for this segment because it adheres to this condensate, and the adhesion energy of this segment because it adheres to the external condensate depleted solution. And already here you see with the blue and the yellow that this is the one that defines the affinity contrast of the membrane to the two different phases. The difference, you can make this affinity contrast dimensionless by rescaling it by the material property of the condensate, which is the interfacial tension. And then by including this force balance equations here, you're going to find out that practically this material property, which is this dimensionless geometric factor, is the intrinsic contact angle, which is the material property, depends directly on the microscopically measured contact angles. And then coming back to this example, if we measure all the three contact angles, we plug it in this formula. For the different microscopic contact angles, we get the same intrinsic contact angle of 45 degrees. I need some water. And here's how the data for the vesicles that I showed on the previous slide look like. We have a dewetting to complete wetting by just looking at this geometric factor. Now, in the next few slides, I wanted to tell you what condensates do to the membranes when they stick to them. When we compare, so here's a vesicle with a condensate, and if we compare the wetted part, so the piece of the membrane which is wetted by the condensate versus the bare membrane, we see that the diffusion in the wetted part is slowed down, and we measure that with FRAP. You see here slowed down by a factor of 2. So when the condensate sticks to the membrane, it slows down the lipid movement. It also changes the packing of the lipids. And the way you can measure that is by using environmental sensitive dye like Lauerdan. The intensity of Lauerdan is not interesting. What is interesting is to look at the spectra, how the whole spectra of Lauerdan changes. And when you do that and use something called phasor analysis and cursor selection, you're going to see that the piece of the vesicle that is in contact with the condensate is much more packed than the one which is than the bare membrane. And this is simply illustrated here with this cartoon. So when the condensate sticks to the membrane, it practically decreases the area per molecule, so enhances the lipid packing and dehydrates the lipids which are in contact with the condensate. If you have a phase-separated vesicle exhibiting, for example, liquid disordered and gel phase coexistence, the gel phase you don't see here, the domains, or liquid disordered, liquid ordered, again, the liquid disordered phase is labeled, you're going to see that the condensate consistently prefers the less packed membrane when it wants to stick. So if you present it with a choice, whether it should go to the gel or the liquid disordered phase, it would always go to the less packed membrane. Okay, so one slide about coupling of membrane phase separation and protein phase separation. This is a little bit crowded, sorry. But the main message here is that if you have phase separation in the membrane, let's say triggered by temperature or raft or domain formation, if some proteins preferentially partition in this domain, you would practically concentrate them locally, and you might form a 2D protein condensate. So this would be a coupling of membrane phase separation inducing protein phase separation. Another example is budding-induced domains, where droplets, when they stick to a membrane which is homogeneous, induce phase separation in the membrane. So induce a domain which is more rich in the blue lipids, for example, compared to the red lipids here in the rest of the membrane. You can also have a local increase in lipid packing, as I showed in the previous slides, and also transmembrane coupling of condensate. You have a condensate on one side of the membrane, maybe a two-dimensional condensate, and the membrane practically induces registration of these two condensates so that they localize in the same position of transversely across the membrane. One more slide that I want to introduce also the word pre-weighting. So pre-weighting is something that occurs below the binodal. So it can occur in your phase diagram where you would expect one phase system, so no phase separation in the bulk of your condensates. But if you bring the membrane next to when you introduce a membrane here, it can act as a nucleation point and two-dimensional condensates can form. So condensation occurs below the saturation point versus weighting where you start already with 3D condensates and you're somewhere here. Okay, and in the last 3 minutes, I want to show you something else that condensates can do. They can do more than weighting, bridging, and deforming membranes. And this is relevant to two of the common cell projects that I developed in our group. One of them is using the photoswitchable lipid azo-PC, which can increase the area of the membrane by shining light. And this is fully reversible with blue light. Here's one example of a vesicle, which is a little bit deflated. It changes its area dramatically when you shine UV light, and then it reverses by blue light, and this can go back and forth. Even if you have a vesicle that is made of gel phase lipids and fluid azo-PC-rich domains, it can induce crumpling, but this is not the point of this project. The point is that you can increase the area of the vesicle, and this with light, and then this can increase the contact area with the condensate. And this is seen with this movie. So you shine light, you increase the contact area, and this is fully reversible with blue light to the extent that with UV light, you can completely engulf a condensate and then spit it out when you shine the blue light in the vesicle retraction area. And one more thing is that condensates can also stabilize vesicles upon damage or upon poration. And the way this was done here is that we encapsulated homogeneous solution of a protein in giant vesicles. So this solution was located somewhere here, for example. And then outside the vesicle, we introduced a solution which is somewhere here in the phase separation zone, which means that if we now make a hole in the membrane and the protein leaves the vesicle, it would mean a solution which would cause condensation, and the condensates would form exactly at the pore. The way we manipulated all these solutions was with microfluidic trapping, where we trap the giant vesicles and then we flow solution after solution. And the way we form the pore is by using a hypotonic shock. So you are practically inflating the vesicles until it ruptures. And here's how this looks like under real-time observation. You have the microfluidic post, the vesicle with homogeneous solution. It deforms, it ruptures, it makes a pore. The whole thing is very dynamic because we apply a flow and the vesicle is rotating. But if you record individual snapshot, you see the pore formation, you see the condensate formation, where the protein concentrates here. And then if you do a three-dimensional image of your pore, you see for your vesicle, you see the hole completely plugged by the condensates. And these vesicles would actually collapse if they don't have the condensate to stabilize them. And with this, I'll stop here. Data that I showed from my group was thanks to these phases here. And I thank also my collaborators and you for your attention. I'll be looking forward to questions.